A-Level Biology 9700, Paper 5, Variant 1, May, June 2024, question number 2 and 3. First of all, we will discuss the question number 2. Question number 2 is the responses of plant species to water stress can be classified as either isohydric or anisohydric. Isohydric plant species close stomata during time of water stress. This behavior minimizes water loss by transpiration, but also reduces carbon dioxide uptake for photosynthesis. So photosynthesis will be stopped during this process when there will be no stomata open. So when they are closed, it means no photosynthesis. And isohydric plant species do not close stomata during time of water stress. This behavior maximizes carbon dioxide uptake for photosynthesis, but also increases water loss by transpiration. So if water is lost by transpiration, ultimately the photosynthesis will also stop. A biologist studies 10 tree species from Australia. The biologist studied the effect of water stress and high environmental temperatures on 5 isohydric tree species and 5 anisohydric tree species. For each tree species studies, the biologist obtained 20 young trees. 20 young trees were taken for the study. The young trees were grown in containers of soil in controlled conditions in a glass house. The environmental conditions in the glass house were chosen to represent summer conditions in Australia. The mean glass house temperature was 28 degrees centigrade. All the young trees were given a good supply of water for 10 weeks so that the young trees acclimatized to the environmental conditions in the glass house. State 3 environmental conditions in the glass house that should be standardized in the 10-week period of acclimatizations other than the temperature of the glass house. So it means during the studies, they have given our mean temperature 28 degrees C and the good supply of water was there. So we have to write 3 more environmental conditions which should be constant during this whole uh, period of study. So the first can be the light intensity. Light intensity. The same light intensity should be given. Same light intensity should be given throughout the study time. Study time of 10 weeks. And the duration of the light intensity should also be the same. Duration to supply the light intensity should also be the same. So this volume of water which was provided should be the same to all plants. It is the same volume of the water should be given. So same light intensity and the duration of light intensity or the volume of the water. So other pH should be the same for all plants. So in this way, we can make our study successful and authentic. Values can be obtained by this using all these conditions in the same way. We are moving towards the next question. First of all, we have to discuss the conditions for question. After the 10-week period of acclimatization, the biologists divided the young trees from each species into 4 groups of 5 trees. 4 groups of 5 trees. You can see the group 1. The young trees were given a good supply of water for 5 weeks. The mean glasshouse temperature during week 1 to 5 was 28 degrees centigrade. So the conditions were same for all 5 trees. The young trees were given a good supply of water for 5 weeks. The mean glasshouse temperature during weeks 1 to 4 was 28 degrees centigrade, whereas during week 5, the mean glasshouse temperature was increased to 35 degrees centigrade. It means only for 1 week the temperature was increased from 28 degrees centigrade to 35 degrees centigrade, but in group 2. The group 3, the young trees were given a reduced supply of water for 5 weeks. Water stress. The mean glasshouse temperature during week 1 to 5 was 28 degrees centigrade. The young trees were given a reduced supply of water for 5 weeks. The mean glasshouse temperature during week 1 to 4 was 28 degree centigrade. So during 5, the mean glasshouse temperature was increased to 35 degrees centigrade. It means the 4 groups in which 2 groups having the good supply of water, but in group 2, the temperature was increased in 5th week. And the other 2 groups, they have uh, reduced supply of water for all 5 weeks. Temperature kept 20 in group 1, 28 degrees centigrade, but in group 2, again for the 5th week, the temperature was increased from 28 degrees centigrade to 35 degrees centigrade. Now, at the end of week 5, the biologist measured the stomatal conductance of 3 leaves from each young tree at 12 midday. Stomatal conductance is a measure of water vapor loss from the intercellular air space of leaves to the atmosphere through the stomata. So the evaporation of water through the stomata, or transpiration rate, you can say in other way. The biologist processed the data to compare the results for the isohydric and isohydric tree species in the 4 experimental conditions as shown in table 2.1. You can see the table 2.1, and here you can see the isohydric tree species and isohydric tree species for the 4 groups. And in the first group, you can say in the water was given, and the other group in which the water was uh, not given. And you can see the both species are shown on this. The bars without color or white color, they are isohydric tree species. They show, and the bars with the color and isohydric tree species they are showing. You can see almost the results are same. You can see from here the stomatal conductance is highest in the first bar, and then it is decreasing. Now we move to the question. Using figure 2.1, state the effect of high mean temperature on the mean stomatal conductance of young trees that were given a good supply of water. So it means isohydric tree species. They are talking about isohydric tree species. It means the bars which are shown with the white color. So we have to, you can see in the second group, when temperature in the first group, the temperature was kept 28 degrees centigrade. But in the group, when the temperature was increased from 28 degrees centigrade to 35 degrees centigrade in the second week, in the uh, last week, in the 5th week. So it has increased 35 degrees centigrade. So in this way, we can see a decrease in the mean stomatal conductance. So the volume was about 470, and it is increased to 400. It is decreased to 400. So in this way, we can say that when we increase, there is an increase in the mean temperature, then there will be the decrease in mean stomatal conductance in these species of the plant. So you can see from this graph. Now we will write our um, findings in the answer. And we can write over here, when there is an increase in temperature, it decreases the mean stomatal conductance. Now, the next question, C part. The biologists then compared the young trees from group 3 and group 4 that were exposed to water stress. Group 3 and Group 4. The biologists carried out statistical tests on the data to see if the difference between the mean stomatal conductance of young trees in Group 3 and the mean stomatal conductance of the young trees in Group 4 were significant. Table 2.2 shows the probability values from the results of the statistical tests, and with the help of you can find out this one, that is between the mean standard conductance of young trees in group 3 and the mean stomatal conductance of young trees in group 4 was significant. So table 2.2 shows the probability values from the results of the statistical tests, and you can see the isohydric tree species, the value of probability, uh, it is 0.046, which is a significant value, and in an isohydric species it is 0.77, which is not a significant value. Now with reference to figure 2.1, which was a bar graph 2.1, this is the figure 2.1 and this is the figure 2.2. Suggest and explain the conclusions that can be drawn made about the effect of water stress and a high environmental temperature or isohydric and anisohydric. You can see over there when there is temperature group 3 and group 4. You can see in group 3 and group 4 the water conductance. In group 3 when water conductance is very low, but in case of this water stress plant it is very low, where in isohydric plant, but it remains the same in both cases in group 3 and group 4, but it is little bit increase or decrease in the values. So we are going to write our results over there in the given space in the light of all these. We have to write about 4 points to explain our answer properly. So with reference to figure 2.1 and 2.2, we will write the answer. So the water conductance decreases mean water conductance decreases in these means in both groups, decreases in both groups. And it happens to save them from dehydration. And when there is, because low conductance low transpiration rate. And when isohydric species are exposed to high temperature, species are exposed to high temperature, the water conductance increases. And 35 degrees, you know, the 35 degrees centigrade is the optimum temperature for enzymatic reactions. When an isohydric plant species were exposed to water stress and high temperature. The results show no change in conductance. So in this way, both of these plants shows their effect differently from one another. And the next question is part D. We are going to discuss part D. The biologists noticed that the young trees in group 4 had some dead leaves at the end of the week 5. The biologists determined the percentage of leaves on the young trees that were dead at the end of week 5. So the trees of types of tree species isohydric and mean percentage of leaves that were dead, they were 19. And standard error value was 8.3 and isohydric 3.5 and the was 1.3. The biologist then analyzed data using a t-test to compare the, and you know students, the t-test is used to compare the values. So t-test the mean percentage of the leaves that were dead on the young trees of the isohydric and unisohydric species, state a null hypothesis for this. So the null hypothesis can be there is no significant uh, differences between the percentage of leaves that were dead or the percentage of dead leaves on young in between the isohydric and species and the anhydric. So we are going to write it this way. There is no, according to null hypothesis, isohydric and anhydric plant species. Now you can see the formula to find out the this formula t-test. We have to calculate the value. X is the mean value and S is the sample standard deviation, and the sample size number of observation, which is represented by N. So we have to find out the value X. The value of X1 is 19.0, where the value of X2 is 3.5. So it is equal to 15.5. So t-test is equal to 15.5 over, they have given this value 8.231. If we divide this, so let me calculate the value. So it is equal to 1.883. So this is the answer of t-test, 1.883. Next part is the degree of freedom for this test are 48. Table 2.4 shows the probability table for the t-test. Using table 2.4 and the calculated value from D to describe what the biologist can conclude from the results shown in table 2.3. So we have to write the answer. For this, you can see the values, and here you can see the value which was table 2.3, the value was 1.883. So the value was 1.883. So we are going to write the answer in the light of this answer. So the calculated value, this is the value of T is 1.883, which is less than 2.011. This is the 2.011. And this is the critical value. It is also less than this value, 2.682. According to these results, we can say that there is no significant difference between this one and this one. Students, you can see that we have to find out that whether it is significant value or not value. You can see that the 1.883, which was calculated in the t-test, it is smaller than this value and smaller than this value as well, but it is larger than this value. So when we are talking about that this is smaller than these two values. So we can say that there is no significant difference between these two values. This value is smaller than these two values. So there will be no difference between whether it is 2.011 or 2.682. So the values are almost very close to each other. So we can say that there is no significant difference between these two values, critical values. You can say that, which is 0.05 and 0.01. So according to this, we can accept the null hypothesis as well. It was all about question number 2. We are moving towards the question number 3. Female mosquitoes feed on human blood. Some species of mosquitoes are vectors of human pathogens. For example, female mosquitoes of the species Aedes aegypti transmit the pathogens that causes the disease yellow fever. So yellow fever, mosquitoes have sensory receptors that can detect chemicals in the air. Mosquitoes repellents contain chemicals that are sprayed onto the skin of humans to prevent mosquitoes taking blood meals. Some scientists investigated the effectiveness of different mosquito repellents using a human volunteer. The scientists carried out the investigation on this person in a laboratory. Figure 3.1 is a diagram of the experiment before the start of investigation. Here is a fan. Its wind speed is 2 meter per second, and human volunteer who is sitting at 1 meter distance. And there are 3 sections. Mosquito cage with net walls, very fine net walls. The mosquitoes cannot pass out, and the thick nets which are center in which the mosquitoes move from one cage to the other. So the same procedure was used for each mosquito repellent studies. A human volunteer sat 1 meter from a mosquito cage as shown in figure 3.1. The person sprayed more mosquito repellent onto the skin of both arms. A fan was turned on so that air moved from the person towards the mosquito cage. The moving air carried chemicals, including the mosquito repellent, from the person onto the mosquito cage. The scientists added 100 female mosquitoes of A. aegypti to section 2 of the mosquito cage. The mosquitoes were left in the mosquito cage for 15 minutes. The walls of the mosquito cage were made of fine net to prevent mosquitoes leaving the cage. The mosquitoes moved freely between section 1, 2, and 3. After 15 minutes, the scientists counted the number of mosquitoes in each section of the mosquito cage. The percentage of mosquitoes section 1 of the was calculated. The procedure was then repeated 3 times on different types days using the same person. Now, state a suitable control for this investigation. Suitable control. So control can be human, volunteer, should without repellent. Or man can spray the water, or man can spray any other oil. Suggest one risk factor to the scientist when carrying out this investigation. So you can see the most risk factor during this investigation can be when the scientist is handling the mosquitoes can bite that scientist. Mosquitoes during handling the mosquitoes. Or any other factor that the scientist can inhale the mosquito repellent, so it can be dangerous for the life of the scientist. Now we are moving towards the next question, and which is the B part. You can see the table 3.1 shows the results of the investigation. Um, Chemical in mosquito repellent DWT 40% and 98% chemical mosquito repellent. You can see in the first DWT it was 40%, in the second D it was 98%, and you can see the percentage mean standard error in both are different. So lemon eucalyptus oil chemical was used only 30%, and its mean standard error is 29.62, which is nearly equal to the DEET, which has having the chemical 98%. So the picardium has only 10% chemical, so its mean standard error is 78.65%. So in this way, the difference is there. Results of this investigation were published in a scientific paper. A student who read this paper concluded that the most effective mosquito repellents contain DWT or lemon eucalyptus oil. So both of they are effective in this way, according to that student. The student was planning to travel to country where malaria is present. The student decided to use a mosquito repellent that contained DWT to prevent malaria infection. Explain whether the procedure and the data in table T.1 support or do not support this decision to use a mosquito repellent that contain DEET. So first of all, I will explain this question and then I will write in the given. So you can see from the table that is 98%. It means it is highly effective at it contains 98% chemical which repel the mosquitoes. So it is highly effective. This mosquito repellent is highly effective, or you can say, but you can say the already told you the mean standard error of both these values, that is the lemon eucalyptus oil and DEET, also they are you can say closely equal to each other. So this is 29.62 and 33.72, which are very close values. So it means both of these can be effective for the against the mosquitoes. But if we compare both of these and the percentage of the chemical which is present in both of these, the lemon eucalyptus oil contains less chemical, and the less use of chemical is highly beneficial for us. So it means it can be more effective as comparison to D, and it is safer. It is safe. It is safe to use. So it contains the less quantity of the chemical. And you can say uh, one more thing which we can say that uh, only 1% was used for this study. If there are more volunteer persons, then the study can be more authentic. So you can say the comparison is more easy if there are more volunteer over there, or you can say there is no statistical data. They have only discussed about this value, about these two values. No statistical data is present. So now I'm going to write all these points which I have discussed with you. So for your convenience, I'm going to write all these. The DEET, which contains is highly effective against mosquitoes. But the values shows that the eucalyptus, lemon eucalyptus oil and beet 98% shows the same or closely values, closely same values. So both of they can be equally effective. But if we compare the chemical point of view, the lemon eucalyptus oil is more effective, because and safe because it contains less chemical. This study can be more authentic if the human volunteer are more in number. And study can be repeated. So students, it was all about the question number 2 and 3. Please like, share and subscribe my channel. Thanks for watching.